We're going to discuss the enzyme structure and the mode of action today. So by the end of this lesson, you'll know what an enzyme is, how does an enzyme work, what are the two theories formed about how enzymes work, and what is activation energy. So what is an enzyme? Enzymes are proteins which are biological catalysts. Now a catalyst alters the rate of reaction without itself undergoing permanent change. It can therefore be used repeatedly and is thus effective in tiny amounts. So enzymes speed up a reaction and remain unchanged at the end of a reaction. They don't make a reaction happen. All they do is simply change the speed of the reactions that already occur. And they do this by lowering the activation energy of a reaction. So we're going to talk about activation energy just now. Two terms that you need to be familiar with are catabolic, which means to break down, and anabolic, which means to build up. Here you can see an enzyme called catalase. It's found in almost all living cells, especially eukaryotic cells. It breaks down something called hydrogen peroxide into water and oxygen. So the enzyme is catalase and the substrate is hydrogen peroxide. Notice how the enzyme's name ends in ASE. So most enzyme names end in ASE, so we call it catalase. So enzymes are globular proteins and they've got an active site where something called a substrate binds. This active site can be made up of only a few amino acids and it forms a hollow depression within the large enzyme molecule. The substrate molecule is held in this active site by bonds. So here you can see the substrate in the blue and the enzyme in the orange. So the active site is made up of a few amino acids and forms a little depression in the enzyme. So the substrate comes along, it binds to this active site. And how it binds to this active site? It binds to the R groups of the enzyme and it forms a temporary bond with the R groups. This structure, the substrate and the enzyme, is known as an enzyme-substrate complex. So how does an enzyme work? Well, enzymes have got this active site that the substrate binds to, and this site is specific to the substrate, as you can see here. So the enzyme's in blue and the substrate's in yellow. The substrate attaches to the active site specific to the substrate. This forms an enzyme-substrate complex. So when the substrate binds with the enzyme, an enzyme-substrate complex is formed. The R groups on the amino acids in the active site interact with the substrate. This lowers the activation energy of the substrate, so it can actually break apart into its products. The enzyme can sometimes change the pH or even have a mechanical tug on the substrate. It'll make it easier for the substrate to break apart. So the enzyme converts the substrate into the products. And notice how the enzyme remains unchanged at the end of the reaction. So here's a little joke. What the enzyme told its substrate: you complete me. If we look at this example, you can see sucrose is the substrate and the enzyme is sucrase in purple. The enzyme sucrase has got an active site that sucrose can bind to. Step 2, where the substrate binds to the enzyme with induced fit. We're going to talk about induced fit just now. And then you have water comes along and the substrate, sucrose, breaks into glucose and fructose. You've got this diagram in your notes. So E stands for enzyme, S stands for substrate. It forms an enzyme-substrate complex. Notice how the substrate fits into the active site of the enzyme, and then it forms the products P. So what are the two theories formed about how enzymes work? Well, the first one is the lock and key, and this was proposed by a man called Fisher in 1890. The key represents the substrate and it fits perfectly into the active site of the enzyme, like a key fits into the lock. Enzymes are therefore specific in the reactions that they catalyze. However, in practice, the process is slightly different. Rather than being a rigid lock, the enzyme actually changes its form slightly to fit the shape of the substrate. It's therefore flexible and like a glove fitting the hand of a person. The enzyme has got a basic shape, like the glove, but this becomes slightly different as it alters in the presence of the substrate. As it alters shape, it puts a strain on the substrate molecule and therefore lowers the activation energy of the substrate. This is called the induced fit theory. So here you can see the substrate in green fits into the active site, but notice how the active site of the enzyme changes slightly when the substrate fits. Then you get an enzyme substrate complex, and the substrate is going to be broken down into the products. So what is this activation energy I've been talking about? Well, if you look here, you've got a substrate, sucrose, and water, so that those are your substrates, and these break down into the products glucose and fructose. Now, for this reaction to occur, the energy of the products, glucose and fructose, must be less than the substrates, which is sucrose and water. These reactions do need a boost of energy to get them kick-started, and this is known as activation energy. It's kind of like a small stone lying on a hillside. If you push the stone, it'll begin to move. If it moves downhill, it'll be better than trying to kick it uphill. So when it moves downhill, it's eventually going to reach the bottom of the hill. And as it's moving, it doesn't need any more input energy. So this shows how activation energy can cause a reaction to continue on its own. So here you can see the graph of activation energy. So if you look here, there's a little energy hill or barrier which must be overcome before the reaction can proceed. So S stands for the substrates, for example, sucrose and water. On the graph you can see that on the very left. Then you've got this little energy barrier that needs to be overcome before the reaction can occur, before P is formed. So what do enzymes do? Well, they lower this activation energy so that the reaction can happen more easily. For example, they allow many reactions to take place at a lower temperature than normal. This means that metabolic reactions can happen quickly in our bodies at 37 degrees Celsius. Now, 37 degrees Celsius is actually quite a low temperature in terms of chemical reactions. So what is activation energy? Enzymes work by lowering the energy of activation. The rate of reaction is increased without the need to increase the temperature. So the presence of an enzyme increases the chances of the reaction occurring by bringing the products into the correct alignment with each other. So bringing the substrate so that they form the products. So for example, the sucrose with the water will be in close proximity with one another inside the enzyme so that they can form the products. And the reaction usually moves in only one direction because of the enzyme. So what have we learned today? We've learned that enzymes are biological catalysts, and they've got an active site that is specific to a substrate. They lower the activation energy of a reaction, and the theory that we now accept is called the induced fit theory. And that concludes our lesson. The end.